whatever going. All right, so welcome back. I hope you guys had a good break. Can you guys see this text here or not? A little bigger? I'll make it one size bigger. How about that? Okay. All right. You still talking about registry? Sorry. So Quiz Friday. Okay. So we got a Quiz Friday. There'll be a help session.
Do you want us to know glycolysis and gluconeogenesis for the quiz, or just
both? Yes.
Yes.
Help session tomorrow at 8:30
I got glycolysis
p.m. to 9:30 p.m. on Google Meet. And remember, if nobody's there, if nobody's there at like 9 or something, or actually I'll only wait 10 minutes in. Nobody's there. This is kind of hard on me doing these things. So if nobody's there, I'm gonna just go. Okay. So I won't be there online ready for people the whole time. Unless people are there the whole time. So I got that help session. I remember my office hours are Monday, 1:15 to 3:15 p.m. and Friday, 1:15 to 2:15 p.m. And then tomorrow I'm going to have a bunch of people in for various things. So let me know if you want to come in tomorrow. I'll be here by 11:30. If you want to make an appointment for some time in there, so I'll be here definitely for 2 hours past 11:30. And I can be here later if somebody lets me know. Okay. That is probably going to change starting next week because my research day has been Tuesday at Virginia Tech, and it's going to move to Thursday. It's based on their schedules as well. So it'll probably, the Tuesday will be more open for times to get together with people. But anyway, let me know if you want to see me tomorrow sometime. The whole day is open. If it was closer to 11:30, that would, after 11:30, but closer to that, that would be better. But I can make other things work as well. All right. Questions? All right. So I handed out some. Oh, actually, let's talk about lab today. So we're gonna have lab today. I think your first lab report will be due next Wednesday, right?
Or it could be not.
Yes. On what, today?
Yeah, actually, it's too cold for my laptop.
I know you're all hoping that. I know you're all hoping we'll get some days off next week, but I don't think that affects our lab. So we'll go with this unless somebody tells me that you guys have on Wednesday some awful test due in some other class.
I think I might.
Yeah, okay. Come prepared next time. So that's when that'll be due.
I think I present for Hollis on Wednesday, so.
Okay, so that's my availability. That's what our lab. And you guys are so lucky. I went up to start prepare lab this morning, and Dr. Chris, who did the lab last year, did it the easy way, and he didn't have any of the materials around for the way I would do it, properly do it. So in other words, in terms of pipetting out your reactions, there are master mixes. I'll talk about that. We'll have to change the protocol slightly. Or there's the pipette out every single thing. Pipetting out every single thing is a much better test and much better practice. It's much better educationally. But Dr. Chris and Dr. Sarisky run it that way, so you're in luck. There will still be some things to pipette out, but not nearly as many. And to your question, Bridget, you'll actually get out earlier because there'll be fewer things to pipette. Anyway, so let's go on here. So we were talking about glycolysis and gluconeogenesis, and what we're going to spend today on is regulation of glycolysis and gluconeogenesis. And what we're going to talk about is we're going to talk about some kind of straightforward or reasoning regulations. So in other words, if I ask you about a molecule, sometimes based on the cell's energy state, you should be able to tell me how it would affect a given pathway. But we're also, in terms of this, we're going to do our big thing for the course on a specific molecule that regulates it. So this is not someplace where you would use reasoning because it's actually a signal molecule that's made. So we're going to talk about both of those things today. When we think about glycolysis, we have glucose. Glucose is going to get made into glucose 6-phosphate, right? All right, and then in a reversible reaction, we're going to take that glucose 6-phosphate, and we're going to make that into what?
Fructose 6-phosphate.
Fructose 6-phosphate. And in a reversible or irreversible reaction, we're going to make that into what?
Fructose 1,6-phosphate.
Okay, and was it reversible or irreversible?
Irreversible.
Irreversible reaction.
Commitment step for glycolysis.
Right, so I'm actually drawing these as double arrows, but maybe I should draw it. Draw these like this, right? Because we're going to have two different reactions there. And then we go on. But this is where we want to talk about. This is the commitment step for glycolysis. All right, so once you get past that, once you have made fructose 1,6-bisphosphate, that is going on to make pyruvate and hopefully have enough oxygen that it's going all the way through the electron transport. It's going through citric acid cycle and through the electron transport chain. Okay, so you go the whole way through. You're at least going to pyruvate at that point. So you could imagine that we've got a couple enzymes here actually first off that are involved. So actually, I'm missing some things. What should I put here?
ATP and AD.
Right? Why don't I write it as ADP plus phosphate? Not Bridget.
Okay.
Because the phosphate that's there gets here added here, right? Okay. And then once again, sorry Bridget, not Bridget. What do I do the opposite direction? You will want this by Friday.
Water to phosphate.
Water to phosphate. Good. Okay. Okay. And how about the enzyme names? Bridget, you can join in if you want.
It's fructose 6-phosphate to 1,6-bisphosphate is phosphofructokinase 1, and then the other one is
Fructose bisphosphatase.
1.
Very good. So phosphofructokinase is going to be a mess. I should have written it underneath. 1. The 1 matters. Or we'll do PFK1 for our discussion here, but you do need to know the whole name. All right. Although if you just remember PFK1, partial credit, right? All right. And then over in the other, we had fructose bisphosphatase.
Mm-hmm. Is it 2 words?
I don't really know.
Yes, it is.
Tase 1 or FBP bisphosphatase, yeah, FBB1. Okay, so we have two different enzymes here that do this. All right, so these enzymes are going to need to be regulated. If one's turned on, the other one needs to be turned off. All right, so we think about that. If we think about for this particular process here, let me do, what do I want to use? I'm going to use black. Let's say I have ATP, high concentration of ATP. What is that going to do for this process? I want you to think about the energy state of the cell and what the cell needs. If if I have a lot of ATP, which of these gets turned on and which of these gets turned off? Going to get turned on down if we have a lot of ATP?
Yeah.
Why do you say that?
It's like, you have extra, like, you can use it, like, the phosphate.
Okay.
to go to-
You're saying it because ATP is a reactant?
Yeah.
Okay. Good reasoning, right? Sometimes that's the case, not in this case, okay? Think about the energy state of the cell, yes.
I was gonna say, if you already have a lot of ATP, you don't need to make more.
Right. This process makes ATP, right? You go down, make pyruvate, it goes to the citric acid site, goes to the electron transport chain, that generates ATP. So if you have lots of ATP, you don't need more. So ATP is going to activate fructose bisphosphatase 1, and it's going to inactivate phosphofructokinase 1. Does that make sense from the perspective of the energy state of the cell? What if I have a bunch of ADP? What does that indicate?
The opposite.
The opposite, right. If I have a bunch of ADP, that means ATP has been used and you need to make more. And so we end up with this. Okay. I want to remind you of something that happens in cells in terms of ATP use. So if you've got, let's say we're talking about muscle cells, heavy ATP use, you get ATP in the muscles, and that's going to get hydrolyzed to make ADP plus phosphate, right? And then what those cells are going to do is there's ways for them to take ADP plus ADP, right? Got a lot of ADP around, and make an ATP. So take one of the phosphates off of one of the ADPs and put it on the other ADP, plus AMP. Like that, okay? And then after that, we have creatine. We'll use creatine to phosphorylate these. And after that, we're using glycolysis to make ATP. There's a lot of other processes that kind of hit in order. After that, you're breaking down glycogen and you're going to fats. And there's a set of orders here in how fast and how easily you can get to the particular energy source. But this is the second reaction here. And so you really don't have a lot of AMP in the cell unless this is going on. And so AMP tends to be a very potent signaler of energy state for the cell. If you have AMP around, what is that going to do? Energy state of the cell. I mean, you need ATP or you don't need ATP? You need ATP. So AMP is going to activate glycolysis to make more ATP, and it's going to inactivate gluconeogenesis. Yes?
I think you need ATP, but you need to use ATP to get that.
Well, glycolysis uses 2 ATP to get started, right? But then it produces, on its own, it produces 4. So you have 2 overall net ATP from glycolysis alone. But then there's another 30 you get if you have enough oxygen and you can make it all the way through. Is that?
Yeah, it just makes sense, but like at this state, like it just doesn't know that. Like it has to use ATP, so it doesn't make sense. Why?
Well, it doesn't know it in terms of the phosphofructokinase, though, has evolved, right? The cells have evolved that this needs to be turned on when there's a lot of ADP and AMP, right? It's an evolutionary natural selection sort of thing. So organisms that had cells that reacted in this way survived more and passed on their genes to their offspring, is the way to explain that. Okay, other questions on that? Good question. Okay. All right. So that is a way we can do this reasoning from the cell, and that a reasoning based on the energy state of the cell, and that's what we're going to do for most of these systems. All right. However, we're also sometimes you just have a signal molecule. Something hits the outside of the cell. It says you need to turn this pathway on. Doesn't matter what the energy state of the cell is, and it's gonna happen. Okay. So if you're the liver cares about the energy state, not just of the liver, but it cares of the energy state of the body and has to respond to that. Yes?
So you wrote down there like ADP plus ADP equals ATP and AMP.
Yes.
If ATP and AMP are both products, why does ATP signal for gluconeogenesis but AMP signal for?
So the question is if I'm making that ATP and I'm making the AMP and they would signal opposite things, right? But there's a lot. So if you think about the way to think about this is that a lot of ATP is like 5 millimolar ATP in the cell. So you could imagine you're getting less. Let's say your ATP, you're depleting your ATP, and this is down at 2.5 millimolar. So you make a little more back by doing this process. AMP, a lot of AMP is like 0.1 millimolar in the cell. But that's a great question, being they're giving different things. So so you don't have to make much AMP to get a really strong response. Whereas in terms of if I took this 2.5 and I raised it up to 2.6, that's not going to say, the cell's not going to say, oh, I've got enough energy now. The cell's going to say it has enough energy when it has 5 millimolar.
Okay.
So that's what I meant when I said it's a more potent, it takes not very much AMP to get that signal sent. Okay, so our signal molecule is interesting because it's very similar to one of our molecules that's in glycolysis. All right, so if we think about this is a fructose, right? We number these 1, 2, 3, 4, 5, 6. All right, this is fructose 2,6, not 1,6, 2,6 bisphosphate. And you can see that based on where it is, where they're labeled, right? Where those numbers are in there. You don't need to know the structure, you need to recognize the structure. Okay, you don't need to be able to draw the structure.
So this is the signaling molecule.
This is the signaling molecule. So this is a signaling molecule. Fonts are just ridiculously small. Give me a second. Look ridiculously small. It's just going to fight with me. All right, hold on. All right, I'm hoping, nope. Okay, I'm just going to do it every time until I figure it out. So this is a signaling molecule. For the regulation of glycolysis and gluconeogenesis. So what I mean by that is, the other molecules are signaling the energy state, sort of, but they're made. It's the cell or the enzymes recognizing what's out there in terms of metabolic products. It is made specifically to turn on glycolysis and gluconeogenesis, and it doesn't have another role I'm aware of. Okay? And that's actually shown down here in terms of if we look at that, look at the step we've been looking at. Is this big enough that everybody can see it? Okay. All right, so we've got glycolysis and gluconeogenesis here. And so this specifically gets made when that body needs, I'm going to say the body needs, because really this glycolysis and this gluconeogenesis aren't happening in all the cells. Really, we want to talk about our organs that are going to be involved in this. Our major one is going to be the liver. Kidneys do this some too, but really we'll talk about the liver. So the liver is either running glycolysis or it is making glucose through gluconeogenesis. Okay, and it's not really doing both at the same time. And so we've got this signal molecule. When this is made, when the fructose 2,6-bisphosphate is made, it's going to turn on glycolysis by activating phosphofructokinase 1, and it's going to turn off gluconeogenesis by inactivating fructose bisphosphatase 1. Okay? But it is not a metabolic product. We don't reason that. And what we're going to do here is we're going to back up a couple steps. We're not going to back up all the way to the cell membrane, or actually we are going to back up all the way to the cell membrane, but we're going to back up a couple steps and then we'll black box it all the way the rest of the way back up. So what causes that molecule to be made? Well, we have another set of enzymes. And this is why we need to be specific about phosphofructokinase 1 and fructose bisphosphatase 1. Because we have two new enzymes here. This is going to be phosphofructokinase 2 is responsible for making our fructose 2,6-bisphosphate from fructose 6-phosphate. Okay? And fructose bisphosphatase 2 is going to be responsible for breaking this thing down back to fructose 6-phosphate. Where are we going to get fructose 6-phosphate from? It's right there in glycolysis, right? So you can pull that right out, make our signal molecule. All right, that makes sense? So why don't I write that though? So phosphofructokinase 2 makes fructose 2,6-bisphosphate. And fructose bisphosphatase breaks down fructose 2,6-bisphosphate. That's essentially what this is showing us over here. And so they need to be regulated for whether we make that molecule or not. Okay? And so we're going to talk about how they're regulated, and then we're going to stop at that level. But we've got a number of steps here that are important in terms of turning these things on and off. And so what happens here is they're actually, I've listed them as two enzymes up above, the figures have, but they're really part of the same enzyme. You've got an enzyme that has two active sites that can do two different things, and one is active if it's phosphorylated, and one is not active if it's not phosphorylated, right? How could adding this phosphate right here cause, at the molecular level, how could that cause one to be activated and one to be inactivated? What kinds of things could be going on there?
Like inhibition of the enzyme.
What was that?
Some kind of inhibition.
Some inhibition. I'm looking for more specific.
The phosphate changes the shape of the enzyme such that one active site can produce the product within two are supposed to off the other active site.
Okay, so people heard that one active, it'll change the shape of the enzyme, right? And one enzyme, one active site could be closed off or whatever happens to it, and the other one could be opened up so it could become active, right? You can think about opening and closing, or it could be you're revealing functional group, whatever's going on there. How about one more level of detail to that? One more detail would just make me so happy. You explain most of chemistry using charges, right? Phosphate's a big charged thing, right? It's a little charged thing, but it's got two charges on it. So you can imagine you put that on there, and the charges of that phosphate are going to repel negatives and attract positives, and that's how we're going to change our shape. All right. So when we see these things get phosphorylated, that's what you should think about. And there are, of course, enzymes here. Well, here we have the enzymes labeled, right? I don't care that you know these enzymes. Okay. What I do care is that you know kind of where this signal is. So we have insulin is going to bind to the outside of the cell, and for now, we're gonna go over a big insulin pathway later. But for now, a miracle occurs. Okay, we actually have a bunch of steps that happen between the outside of the cell, and that activates the enzyme. We just say the enzyme that's going to cause the phosphate to come off. It's a phosphatase, right? I hope you're recognizing, or you're starting to get used to these terms here. They are in your study guides. I haven't emphasized it. What does a phosphatase do?
It takes away a phosphate.
It takes a phosphate off, and a kinase puts a phosphate on, right? And a kinase really specifically from ATP, usually, right? Sometimes these enzymes are named, if we're looking at a pathway, sometimes they're named for the reverse direction, even though it doesn't happen much, because that's how it was discovered. Sometimes you've got something called a phosphatase, but really, when we talk about it, we're usually talking about it as a kinase. It's catalyzing the reaction both ways. But in general, a phosphatase is going to take something off, and a kinase is going to put it on. Okay, so what do you need to know here? Insulin is going to cause the enzyme. Let's go ahead and write it out. Actually, we'll make it slightly more specific. Insulin causes the phosphatase. I don't care that you know the name. That removes the phosphate from the PFK FPBase to enzyme, we'll just say enzyme, because it's not really a complex. Let's make that a removes. To be activated. This removes the phosphate. I'm going to copy it again. So our phosphofructokinase, fructose bisphosphatase enzyme, 2 enzyme, changes shape, causing phosphofructokinase to become active. The PFK2 active site to become active. I'll spread this out. And the FPBase2 active site to become inactive.
So they regulate the regulator?
Yes. And we could follow this back, and you could have you regulate the regulator of the regulator, you regulate the regulator of the regulator of the regulator. We have things like that. It is silly, but it's how these things work, and it's not. I mean, it works. So it's not silly that it works that way, but it is kind of silly to talk about sometimes. All right, let's. So the insulin's going to cause this it's going to cause this phosphatase to become active. It's going to that's going to remove the phosphate from our enzyme here, okay, which is going to cause its PFK2 active site to become active, and we're going to make fructose 2,6-bisphosphate, which is going to activate glycolysis. Does that make sense?
So basically, insulin makes the F2/6.
Insulin activates glycolysis.
Okay,
Yes?
because it makes the F2/6.
Yep. So then right here I'll say fructose 2,6-bisphosphate is made, right? It binds to, right, at a molecular level, if we got a molecule that's going to cause these two enzymes to change their activity, and now I'm talking about fructose 2,6-bisphosphate. So which enzymes does it cause their activity to change? Yeah, the ones, right? The ones. We're now back to the ones. It binds to PFK1 and I'll have to move that out of there, and fructose bisphosphatase 1, activating the first and inactivating the second. Let's stop and look at that. Do people have questions? Because I think I'm not going to write. I'm getting tired here. I don't think I'm going to write out the opposite case. The opposite case is we'll talk about it. We can talk through it. But yes.
Can you draw arrows to where your ones are talking about?
So where are my words on the upper things? Okay. Happy to do that. So let's.
So just like the paragraphs.
Yeah. So this paragraph right here, right? Actually, this part is going to take us. I don't know if this is going to be helpful or not. Actually, let me try shrinking it and see if it how helpful it is if we look at it right here. All right, so that bit right there that says fructose 2,6-bisphosphate activates and activates is where we're back at the ones, right? So this is here. Okay. The first sentence says fructose 2,6-bisphosphate is made, right? That bit is here. Okay. Does that help?
Yes.
That's a great suggestion. Other questions? Okay. So let's just talk the reverse. I'm going to talk through the reverse process, but I'm not going to write it out. But it's the reverse process. So what we have here is, so we have this other enzyme that's a kinase, glucagon. All right, glucagon signals you have low blood sugar. Okay, like I had before I started eating a sandwich before class. But it signals you have low blood sugar, and it's going to cause the phosphorylation of this enzyme complex. Okay, when it's phosphorylated, it inactivates PFK2, and it activates fructose bisphosphatase 2. This is going to tear down fructose 2,6-bisphosphate. So it's the reverse of this. That's taken away, and that's going to result in glycolysis being turned off and gluconeogenesis turned on. Is that good enough? Okay, so that's the reverse process there. You should be able to step through that, write an explanation that steps through that. Okay, so from here, talk about a kinase being activated, talk about a phosphate being put on, one of these being activated, one of these being activated, right? The one that tears down fructose 2,6-bisphosphate being activated, it's going to tear it down, our signal molecule isn't around anymore, and we no longer have glycolysis going. Sophia, did you have a question?
I think I was like processing the fact how insulin you have to go from like the back like H2O to the phosphate, but then and that makes the fructose 2,6 bisphosphate, which like inactivates that pathway.
It's activating the making of fructose 2,6-bisphosphate as opposed to the tearing down of fructose 2,6-bisphosphate. That's what this one does.
Yeah, but then so then that okay, then what does that do again? K2.
So no, that's fine. Let's do it. Let me go ahead and add another. So this right here breaks fructose 2,6-bisphosphate down.
Okay. And the other one makes it.
Yes. Okay, and then here, this is going to be makes fructose 2,6-bisphosphate.
And so can you explain and then why does it activate the ATP, the ADP way instead of the H2O to the phosphate?
Why does insulin?
Why does fructose 2,6-bisphosphate? I jumped around. Like,
Here? In this figure?
Here, like in the beginning after we've already made the F26BP.
Right. So here.
Yeah.
Why does this activate this?
Yeah.
It's going to bind to it, cause a shape change.
Okay.
It's going to all these things are going to be bind, cause a shape change. And so this will become active. And then it's going to bind also to our fructose bisphosphatase 1 and cause a shape change and shut it off. And so then when this is removed, it's binding non-covalently. So when this is removed, it can come off because there won't be much in solution. It's Chatelain's principle, right?
Mm-hmm.
It'll come off, and then this will become active, and this will become inactive.
Okay, yeah.
Good questions. Other questions before we move on? Okay. Then let's go ahead and we're gonna move on. We're almost at the citric acid cycle. We need to talk about the step in between glycolysis and the citric acid cycle, which involves the pyruvate dehydrogenase complex. So to remind ourselves, we start out with polymers, right? Actually, let's be specific here to the polymers we're working with. So we got glycogen. We're going to have glycogen breakdown, or we'll make it. Then we have glucose. And then we have glycolysis and gluconeogenesis. And then we end up with pyruvate, right? We have 2 or 3 carbon intermediates, but it's a 3 carbon intermediate for pyruvate. And we need to make that into CoA, right? We need to actually make it into an acetyl group that goes on to CoA. And so we're going to come through what's called the pyruvate dehydrogenase complex. And so this will be acetyl-CoA. And we have the pyruvate dehydrogenase complex. All right, and coming into this, we're going to have a CoA. We'll talk about the structure of CoA in a minute. All right, and actually coming out of this, we're also going to be per pyruvate, we take one NAD plus and we make an NADH. All right, so per glucose, how many NADH do we get out of this step right here? 2, right? Because per glucose, going through going through glycolysis here, we would get 2 pyruvate out the end, so we'll get 2 acetyl-CoA. Questions? Okay, and then what we're going to do, now that we have our acetyl-CoA, now, actually, I draw it like this. We're going to go into the citric acid cycle. I'm just going to do CAC for this right here. We're going to end up with 2 carbon dioxides out of it. We go in with 2 carbons with a CoA. You're going to end up with your CoA coming out, right? You end up with those. And then energy-wise, the important things is we end up with 3 NADH. That's most of our energy. We end up with 1. I've said FADH2. I'll also say ubiquinol. Okay, I'll show you how they're related. You may have seen FADH2, you may have seen ubiquinol, depending on whose figure you're looking at, and then we end up with 1 GTP. All right, and this is per acetyl-CoA coming in. So I'm going to go back up here, I'm going to erase these 2's. Because you get double that per glucose. Does that make sense? Okay, so then we have our electron transport chain. At the start of our electron transport chain, we're going to end up with our NAD plus entering our FADH2, or it's now ubiquinol. Our ubiquinol is going to actually enter a little bit later. And we're going to have oxygen on this side, and we make some water, we pump some protons. Those protons are going to come back through that ATPase. Or I should say ATP synthase in this case. And through that ATP synthase, we're going to get ADP plus phosphate. The protons coming back in are going to power making ATP. I'll just go ahead and put a membrane here. So this is my electron transport chain, and this is ATP synthase. Okay. So we've seen that before. You guys did pretty well on that quiz for that. But the important point here is this: you are here for what we're talking about. We're at this point in the process. We're going from pyruvate, turning it to acetyl-CoA. And while I haven't drawn arrows for it, this NADH is also going to go to the electron transport chain. All the NADH, an NADH made in glycolysis, that also goes to the electron transport chain. That's where it's all going. Okay. We call this the pyruvate dehydrogenase complex because it's the complex of a bunch of different enzymes. So this is I don't know if it's electron, I don't know what kind of structure this is, but probably not NMR. But this is the pyruvate dehydrogenase complex. All right, it has 3 enzymes in them. I think they're labeled E1, E2, E3. I don't care that you know that. They're actually colored that way here, but you can see that you have more than one of each, right? We have at least 3. I'm not sure if we have 5 in some of the back. Maybe it's just 3 of each type of enzyme here in terms of that complex coming together. All right, we're going to talk about how the reaction works here and the cofactors, the enzyme cofactors that are involved. But we're not going to concern ourselves with like how many of each there are. They'll kind of act independently. As far as I know, they're not cooperative. All right, but here's an example of E1, E2, E3. I don't care that you know that they exist even. But what we're going to do is I'm going to walk through kind of how this reaction happens, and then we're going to talk about the specific cofactors involved, and then we're going to walk through it again, okay, in terms of that and understanding what's going on. So we start out where we have a pyruvate right here. The pyruvate's going to interact with a cofactor called thiamine pyrophosphate that we'll see in a minute. Okay, and we're going to lose a carbon dioxide right there, right off the bat. I should have shown a carbon dioxide being lost in my overall picture. And that 2 remaining carbons is going to be captured as this hydroxyethyl group. Okay, that's on the thiamine pyrophosphate. That's going to come over here, and that's going to be handed off to a lipolysine or a lipolate. I like to call it a lipolate. The functional group is a lipoate or lipolyl or whatever. The lysine bit is it's attached to a lysine. Okay, so it's been added to you've modified a lysine to attach it to it, and it has this thiol on the end, right? So if I look at a thiol and I think about reduction, let's compare this thiol to this thiol. Which one is reduced? The one on your right or the one on your left?
Right.
This one's reduced. Okay, so we got this oxidized. We're going to bring it in and we're going to reduce it, right? Or at least partially reduce it here, where we've got now an acetyl group attached, and we now have a reduced lipolysine. Okay, so we had some oxidation that's happened here. You can see that oxidation has occurred, right? Because
So, ketone now.
yeah, we had a ketone there, but we had 2 hydrogens. We lost 2 hydrogens. So oxidation occurred here. This became reduced. That's awful. What's wrong with the way they drew this? This cartoon. I like this cartoon. Got a bond. Looks like the bond's going to the H. Should be going to the S, right? But anyway, besides that, it's a good figure. So, and then this is going to, the lipoate is going to swing around, or the lipolysine, I don't care what you call it for this. It's going to swing around and it's going to hand off this acetyl group to CoA. Okay, and now we've made an acetyl group. It's a 2-carbon intermediate, and we've attached it to a CoA, and it is ready to go into the citric acid cycle. So this goes to the citric acid cycle. All right. But now there's a problem, right? To make an acetyl-CoA, what form does my lipolysine need to be in? Lipolysine, lipoate.
Oxidized.
It needs to be oxidized, right? So we need to reoxidize it. Okay, so this needs to be regenerated. So the enzyme can act again, right? Enzymes aren't good. They don't just do once. Actually, there are some examples of that, but and they still get called enzymes for some reason. But mostly an enzyme is going to be regenerated, so it can do this process all over again. Plus, it has an NADH's worth of energy in there that can be captured. So we want to get that energy out as well. So what's going to happen is this is going to swing around, and it's going to hand off its electrons to an F8 along with hydrogens to make an FAD, take an FAD and make it into an FADH2, okay? And that's going to regenerate our oxidized lipolysine. All right, and then FADH2, I may have mentioned this already. I'm going to mention it a couple other times. FADH2 is always tightly bound to enzymes. And how much energy it can capture is dependent on the environment it's in the enzyme. So here, this FADH2 is capturing at least how much energy? And you don't have a full feeling for this yet because we haven't counted ATP yet. But an NADH is worth a lot of energy. It's worth about 2.5 ATPs worth of energy, depending. But NADH is worth about 2.5 ATP. So that means that this FADH2 in this context is carrying at least 2.5 ATPs worth of energy. If that makes sense. Because it's going to get used to make an NADH. All right. So it's going to hand off its hydrogens plus the electrons to NAD plus to make NADH, and then this can float off to the electron transport chain.
So what of this do you want us to know?
Oh, all of it.
Unfortunate.
Sorry, for the post Friday.
We can push it off to the next quiz, I
Thank
guess. Yeah. What is it? Wednesday? Yeah. Let's just have to hope for snow. Is it Friday or is it Monday? Which?
It's supposed to start Saturday.
Oh, that's not going to help you for Friday.
Wait, what
Yeah, but if I don't have to know this,
What are we supposed to know for the quiz? Just like glycolysis?
everything up
All
until
the past, like,
today. Yes.
the pentose phosphate tablet.
Do you want it all? Do you want it all surprise on the first test?
I mean, I can only learn so many mechanisms at a time.
You know,
okay, wait,
past
can
classes have learned all this.
Can you list, okay, so like glycolysis, gluconeogenesis, pyruvate dehydrogenase complex.
We can go back. We can go back and do that. All right.
Yeah,
So
email. Email works.
you're going to have to remind me on that because I'm going to leave here. I'm going to scarf down that sandwich and I'm going to frantically try and get the rest of the lab prepared upstairs.
Fair enough.
So I can do that in email, yes, but I'm going to need a reminder. It may happen during lab, but it's not going to happen. Well, send the email. Okay.
Wait, so that last bit that you just went over is on this Friday?
It's not on this Friday's quiz.
But we don't know the E stuff about it, but the actual topic.
Okay.
You need to be able to describe what's going on with these various
And
cofactors.
draw it.
I don't know that you mean. I've never asked anybody to draw it.
Just like stuff that was going on.
But we're going to talk about the cofactors here.
Right.
Okay. So this is vitamin B1, I think.
Yeah.
Thiamine pyrophosphate is vitamin B1. Okay. If you're low on this, you end up with things. There's a disease called beriberi. Something that sailors used to get this because they didn't get enough vitamin B1. People who are alcoholics can get this because you have trouble getting enough vitamin B1. But it causes problems with energy, right? Beriberi, in whichever language it comes from, means I can't, essentially, because you're tired out. And it's because you can't run, you can't make acetyl-CoA, so you can't take all your glucose and turn it into carbon dioxide and get all the ATP out of the electron transport chain, right? Because you don't have this cofactor that's needed to make the acetyl-CoA. All right, so this is thiamine pyrophosphate right here, and we have two forms here. This is the unloaded form. So we talked about oxidized and reduced. We now have a loaded and unloaded possibility for our cofactors. So what you need to do with this is you need to be able to, this is our recognized, is it the loaded or unloaded form? So you recognize that you name it, loaded or unloaded form, and then draw the other form.
For this Friday.
No, not for this Friday.
He said that those are going to be on Friday.
Wait, what does loaded and unloaded mean?
I'll go over it again. Recognize, name, Name, identify as loaded or unloaded. Draw the other form. Okay, and I mean all atoms when I say draw the other form. Okay, you can use line angle when I say that. All right, so what's the loaded versus unloaded? Sophia disappeared. Okay, so loaded versus unloaded. This is unloaded. All right, this is unloaded right here. And this is the one that has the hydroxyethyl group on it. It's got the 2 carbons that are being carried along, right? Loaded. So here are 2 carbons, right? First step of the pyruvate dehydrogenase complex is you take pyruvate, you get rid of the carbon dioxide, and you capture these 2 carbons. And they're going to be loaded onto acetyl-CoA in a couple of steps. Questions? Yes?
So is this the reduction, I guess, or the oxidation from
This-
pyruvate to?
This is I would say this is going to be reduced here, or you released a carbon dioxide, right? So we're getting the reduced bit here. You oxidize the part that left. If that makes sense, is the way I would think about it. But it's got to be a reduced thing we're carrying along because we're going to use it to oxidize. Well, we're not going to use it to oxidize. We're going to use it to reduce the lipowave and then capture some energy there. Other questions? Yes.
Why does it use pyrophosphate here and not diphosphate like ADP?
Why does it not have an ADP handle, you mean? Or
Isn't it also like a diphosphate on the end? It's called a pyrophosphate.
It is. It is. So why do they call it pyrophosphate instead of diphosphate? I don't know. It's historical.
Yeah.
Okay, I'll see you guys in lab. We'll go over there are 4 more cofactors we'll go over on Friday.